Okay. Algal nitrogen preference. Basically, all right, so phytoplankton can, for nitrogen, phytoplankton can utilize ammonium and nitrate. All right, in model world, they utilize one of two things, ammonium and nitrate. In the real world, in the presence of almost any ammonium, they will use that first. There's an it has to do with energy and algal physiology. It takes less energy for algae to use ammonium than nitrate, all right? I won't go further than that. So we have a preference, what we call a preference function, that looks at the amount of ammonium that's out there, the amount of nitrate that's out there, and comes up with something called a preference. And the preference is the fraction of the total nitrogen that algae take up, what fraction is ammonium? And it can vary between 0 and 1. Take a look at this. You see who's been around since 1982, right? And Jim, that is, this is a modification of something that Jim and Bob Toman came up with 37 years ago, something like that.
Little kid in diapers.
What's that? There you go. Okay. In all seriousness, this is a modification of something that Jim and Bob came up with. When they did it, this K, this was the half saturation constant, that Monod constant for nitrogen uptake. And when I and some other people looked at that, what we saw was that the preference for ammonium was probably not as high as it would be. So in ICM, we have we specify something here, it's called KHNH4, and that is a specific half saturation constant for ammonium.
So that'll be lower than the regular nitrogen.
There you go. That's exactly right. And so if you look at the that function, what you see is that two things influence the preference. One is the amount of nitrate relative to that KH, and that's shown on this axis. And then the other is the amount of ammonium relative to KH, and that's kind of shown by these different lines. But as you might expect, as the amount of nitrate diminishes, the preference, if you will, for ammonium increases, all right? So as nitrate decreases, and then as the amount of ammonium relative to the half saturation decreases, here the ammonium is what, 10 times the half saturation. So the preference, as the ammonium diminishes, the preference decreases. So if you've got a case where clearly, if there's no nitrate, the preference for ammonium has to be 1 because there's no nitrate. It kind of levels out, and then it becomes more or less influenced by, if you have abundant nitrate, if it's really a lot of nitrate, then the ammonium preference is influenced by the abundance of ammonium. If you've got a lot of ammonium, even if there's a lot of nitrate, I'm going to use that ammonium first. If you don't have a lot of ammonium, well, I'm hungry, I'll take what I can get, so I'll use some nitrate. Okay, so this is a parameter that you're going to want to specify, and as Jim mentioned a few minutes ago, it should be somewhat less than the half saturation concentration for nitrogen uptake.
So just out of curiosity, was that, you made that modification because of information in the literature, or because it really improved the calibration of the ammonia and nitrate in the model?
Literature and criticism from reviewers.
Oh.
I can't, they picked that out. I'm not going to claim it made the model better or worse,
okay,
but those were two influences.
Okay, thank you. I mean, I guess at the time, not sure what the literature was, but I guess the other thing that it does, thinking of how Bob's philosophy was, don't add more constants than necessary. You know, so you're adding another constant that you need to specify. Okay.
Let's see what we got. Okay, so I don't know if I'm going to get into the weeds there. There are a number of things. I don't know how far into the weeds I want to go, and I'm just going to go over them quickly. There are algal species that like fresh water but not salt water, and there are algal species that like salt water but not fresh water. Recall that what I said was, in this model, we allow for 3 algal groups or algal species. And in Chesapeake Bay, we have called those 3 groups freshwater, spring bloom, and everything else. And you are allowed in the model code to specify all different parameter sets for 3 groups. And as I said, in the Bay, those are the 3 groups that we define. If you were applying this to a different system, you can make them anything you like. Or you could only run one of them. If you say, I just have general algae, I'm not going to worry about 3 groups. We have in here, and I won't go over this in detail, but we have some salinity toxicity functions in here. So you can, through the model parameter set, not let that freshwater algae group grow in salt water, or you can, through the model parameter set, not let those saltwater algae grow in freshwater. Actually, it's not a restriction on growth, it's a mortality. When those freshwater algae get washed downstream into salt water, they die fast, and vice versa. So I'm not going to go in there. I'm just going to tell you that that facility is available, and we'll stop there. Apparently,
can I ask?
Yeah, go ahead.
A question? You don't have a blue-green algae.
Yes, those are the freshwater. They qualify as freshwater.
They're freshwater.
That's correct.
Okay.
We used to actually define that as blue-green algae. In earlier versions of ICM, it was defined as blue-greens, and it was specific to the Potomac River because that's where we have the blue-green algal blooms. But it turns out there were other freshwater regions where, you know, the algae seem to have distinct properties even if they're not blue-greens. The Patuxent is one. So I just call that generally freshwater algae, and you might, if you had the data, use a parameter set in the Potomac that would represent blue-greens, and in the Patuxent you might represent freshwater diatoms, you know. Okay, the parameters, not all, but several of the algal parameters are spatially variable. So you have 3 groups, fresh, diatom, and other. But I could make the growth rate of freshwater algae in the Potomac and the Patuxent different. They're spatially variable.
Oh.
So there you have it. Like I said, if I think I have one group in the Patuxent and a different group in the Potomac, I can make those variable.
They are spatial, but in the current model, they are not spatially variable.
I think they are, in fact.
Okay.
As I mentioned, I know the Patuxent and the Potomac are different.
Gotcha.
Okay, so we've been through, we've talked a little bit in detail about phytoplankton. Now let's look at some of those other cycles. This is the nitrogen cycle. And once again, as with the carbon, if you look at the cycle, if it's got a box around it, it's a model state variable. Probably a difference between, distinction between the nitrogen cycle and the carbon cycle are that the carbon cycle was open, which is to say we had that dissolved inorganic carbon that didn't have a box around it, and we just said if something goes there, we forget about it, and if something comes from there, we assume the reservoir is infinite, okay? So there's nothing here that doesn't have a box around it. So for nitrogen, we're very careful to conserve nitrogen, okay? There's no infinite sources or sinks where we can just get as much as we want, or it goes there and we don't worry about it anymore. So model state variables: ammonium, nitrate, dissolved organic nitrogen, the three alkyl groups. Okay, the reason they appear here, remember, is what we said, that we have to keep track of the nitrogen that's associated with the carbonaceous biomass, and then we have the three particulate organic nitrogen groups. We'll speak about, well, let's talk about those for a moment. Those particulate nitrogen groups, they correspond to the G1, G2, G3 that Dominic talked about yesterday. So we have a labile, a refractory, and you could call it slow refractory, you could call it inert, you could call it G1, G2, G3. I think in the model documentation we use labile, refractory, and G3. Now I don't know, well, let's just wade into the weeds there. The decay rates in the water column can be different than the decay rates in the sediment, okay? But, you know, on the whole, order of magnitude type thing, they're similar, okay? I'll mention, I don't know, earlier versions of ICM only included in the water column labile and refractory. And when we took that refractory material and deposited it in the sediment, we split it into G2 and G3. And that worked for a while, but what we found in this model application was that different sources of nitrogen, sources meaning loads, had different compositions. And we could no longer just assume that we had the sort of general refractory nitrogen that we could split into G2 and G3. What Jim was reminding me of last night, I don't know how many of you are familiar with the Conowingo Dam right now, but there's a big issue in the Bay community about scour. The reservoir is full, the bottom scours and dumps immense quantities of nutrients into the bay. What's that bottom made out of? You know, how labile, refractory, or inert is it? And a lot of work that Jim has led has been devoted to measuring the fractions of reactive material in that bottom sediment. And given the work that went into measuring those fractions, we wanted to represent very accurately in the model the loads. We have a similar issue with shoreline erosion. There's a lot of nutrient, particularly phosphorus, in shoreline erosion. And once again, that material has specific fractions, and we want to represent those loads as accurately as possible. So for this phase of the modeling, we specifically added G3 so that we could, for the different loads, we could, to the best of our ability, represent how those loads are fractionated and have those correspond to the sediment variables. The ammonium, so that said, ammonium undergoes a process called nitrification, where it's converted to nitrate. It consumes dissolved oxygen along the way, very important. The three alkyl groups, as we mentioned, they take up ammonium and nitrate, and you know, the fractions that are withdrawn from either of these two pools are determined by that preference function. When the algae die, they're partitioned into a lot of substances: ammonium, dissolved organic nitrogen, and those three particulate groups. So you as a modeler are faced with rooting this dead algae into the, what are there, 1, 2, 3, 5 different groups. And again, if you do this for years, you get a feel for for where, you know, some idea what those fractions are, but it's one of the model tuning things, and it can be very important to your calibration how you partition those. The particulate organic nitrogen groups hydrolyze to dissolved organic nitrogen. The dissolved organic nitrogen mineralizes to ammonium. And, you know, the rates of hydrolysis and the rates of mineralization are model parameters that you specify. These are key. And once again, if you do this for a long time, you have some sense of what those numbers ought to be. But, you know, these are key model calibration parameters, and when you're going through trying to apply the model, these are things you really want to be careful with. Or if not careful, you're going to devote a lot of attention to these. And they settle to, the particulate groups settle to the bottom. And once again, we saw the diagenesis model yesterday. It's also possible for nitrate to diffuse into the sediments. In fact, it does. And once again, that's accounted for, or that involves the diagenesis model. Let's see, what else do we have?
Carl, for back slide, the back one.
Go back.
Yeah, and I want to look in this your figure that I have a question. Does denitrification happen in the water column?
Oh, what a question!
I'm trying to find it, but not, you know, that's.
We're gonna get a lot of controversy about this. Okay.
Okay.
Denitrification is, in the absence of oxygen, nitrate will be reduced to nitrogen gas. And that consumes, you know, it takes energy to do that. Bacteria do this. And the energy comes from carbon. So it's a carbon sink. I can't write the equation on the board from memory, but we saw it yesterday in our lecture on the diagenesis model. There was denitrification. So very many eutrophication models, including this one, have denitrification in the water column when the oxygen goes to zero. And until recently, that process was represented in our model. Okay, and in fact, the code is still there. But we parameterize that so it does not occur. And, okay. I actually went out and I said, okay, you know, we're denitrifying nitrate. Let me go out and find some measurements. I've never found one for Chesapeake Bay. So if this is an important process, how come nobody's measured it? And I kind of went and looked and looked and looked, and I found a paper by Sybil Seitzinger about denitrification in the water column, and it turned out it's a rare process that only occurs under very, very special conditions.
Well, you know what it probably is? It's a relic before the sediment flux model was there, because nitrate was disappearing.
Yeah.
So that was our, being a modeler, that was our rationale. The nitrate's disappearing. We weren't modeling sediment flux.
It could be.
And it's because the oxygen is not present and getting into the sediment, so therefore that's the source of oxygen for the sediment, so the nitrate flux was in. So I think that's what it is, and we just never really thought about it and turned it off. So you have turned it off.
So, you know, what we've done, so the kinetics are there, but it's parameterized such that it doesn't occur. And, you know, I mean, I never thought about it. It just seems natural. You know, denitrification's a process, and there's no oxygen, must be going on. And everybody else does it, right? So, but as I said, when I really tried to find measurements that would justify the parameterization in the model, I mean, what should the denitrification rate be? I had a tough time finding those measurements, and when I found them, you found out that it's not a common or universal process in the water column. It's very rare. So I wish I could find, I should have spent time finding that paper before the class just to distribute it, but I'll just say, so I won't get on the soapbox and I won't tell you anything. I'll just say in the Bay model right now, we don't consider it. And enough said, you know. I think I was going to mention that. Richard beat me to it, but anyway, okay.